Hello everyone. Today I will introduce our recent work, which has been submitted to iMeta, entitled Distinct Microbial and Metabolic Shifts Characterize Acute Coronary Syndrome and Recovery. This study was carried out in collaboration with Fuwai Hospital, the University Medical Center Groningen, and other partner institutions. Coronary artery disease is the leading cause of death worldwide, and acute coronary syndrome is its most fatal form, often caused by plaque rupture. While gut microbes and their metabolites have been linked to CAD, ACS-specific signatures remain unclear, especially in distinguishing it from stable CAD. Since early detection of ACS risk in sCAD patients is a major unmet need, our aim was to identify microbial and metabolic markers that separate NCA, sCAD, and ACS, and to track their changes during recovery. Our results revealed stage-specific alterations in gut microbiota and metabolites across NCA, sCAD, and ACS. Many ACS-specific features showed partial reversion after clinical recovery. Multi-omics machine learning models accurately distinguished CAD subtypes, significantly outperforming models based on clinical markers alone. We studied 548 participants, 175 with normal arteries, 161 with sCAD, 212 with ACS, and 52 recovery patients. All had fecal metagenomics and plasma metabolomics. Compared with controls, sCAD and ACS patients more comorbidities and higher inflammatory and cardiac markers, while lifestyle and diet were similar across groups, as shown in Figure S1. In Figure 2, ACS patients had higher Simpson diversity and clear species-level separation from NCA and sCAD, while pathway differences were weaker. They were enriched in pro-inflammatory, oral origin, and mucin-degrading species, with a lower Prevotella to bacteroids ratio. Additionally, ACS showed enrichment in lipid and inflammatory metabolism pathways. In Figure 3, metabolomic profiles separated NCA, sCAD, and ACS. ACS patients showed elevated organic and carboxylic acids and phenylpropanoic derivatives, but reduced amino acids and neurotransmitter-related metabolites. Lipidomics revealed higher triglycerides and sphingolipids, with lower phospholipids and fatty acids. Pathway analysis highlighted amino acid and carbohydrate metabolism reprogramming linked to inflammation. We then focused on ACS-specific changes. ACS patients had more streptococcus and bifidobacterium, but fewer clostridium and desulfovibrio. They were enriched in pathways for ketogenesis, fatty acid, and bile acid metabolism. Metabolomics showed higher 2-hydroxybutyrate, phenylalanine derivatives, and succinate, with lower lysine and A. Lipidomics revealed more triglycerides and diacylglycerols, but fewer phospholipids. Features correlated strongly with NT-proBNP and hsCRP, linking them to inflammation and cardiac injury. After identifying ACS-specific features, we asked whether these changes reverse upon recovery. We analyzed 52 patients who moved from ACS to a stable condition. Their profiles resembled sCAD and diverged from ACS. Pro-inflammatory taxa such as streptococcus decreased, while ACS-elevated metabolites like 2-hydroxybutyrate, 3-hydroxybutyrate, and succinate declined, and several amino acids increased. These results suggest that ACS-related microbial and metabolic changes are partly reversible, making them potential biomarkers for monitoring recovery. Given the metabolic alterations in ACS, we next asked whether these metabolites are linked to gut microbes. Using MET-origin to integrate metagenomics and metabolomics, we found that 2-hydroxybutyrate, 3-hydroxybutyrate, and succinate were elevated in ACS and positively correlated with multiple microbial taxa. In contrast, tryptophan was reduced, negatively associated with certain microbes. Gene analysis confirmed enrichment of microbial genes for 3-HB, succinate, and tryptophan metabolism, suggesting that gut microbes may contribute to ACS through metabolic regulation. We asked whether these signals could discriminate CAD stages. Clinical markers alone performed poorly, but adding microbial and metabolite features improved accuracy, with AUCs up to 0.91. Overall, the multi-omics models outperformed clinical markers for risk stratification. In conclusion, we showed that ACS patients have distinct microbial and metabolic alterations, many linked to inflammation, with partial reversal during recovery. Integrating multi-omics data allowed better discrimination of CAD subtypes than clinical markers, especially for ACS. These findings highlight microbial and metabolic features as promising biomarkers for risk assessment and potentials for intervention, offering new perspectives for precision management of ACS. Thank you.